Okay, so today we are wrapping up the single cell section. So last time we talked about both single cells and spatial techniques, and we talked a little bit about how to process single cell data. So this week we are going to focus more on the data analysis, like what biological knowledge can be learned from single cell. So we can do a quick recap of the QC and batch effect removal that we mentioned last time. And today we're going to focus on the visualization of single cell data and how can we infer cell type and how can we infer the trajectory or the developmental trajectory of cells from the data. So last time we mentioned a little bit that once we got the single cell, like we have to look for, we have to perform some quality filter. Like some barcode might correspond to empty droplet, like there's no cell inside. Some barcode might correspond to multiple cells being incorporated into the same droplet by chance. And some barcode might correspond to dead cells. And we don't want for these to confound our analysis. We talked a little bit about how we normalize and impute the missing measurement in the data. And then we went over the how we integrate sample across batches. And last time I kind of highlight this that some genes can affect the visualization and clustering. So today we're going to mention this, how the decision of which gene to consider in your analysis can impact the result. But let's start with the key techniques first. The first one is how we visualize the data. So since last time we saw scatter plot like this, where each dot is a cell, and we probably some of you probably question like how can we like visualize or project single cell data onto the screen, because each cell is described by expression level of more than 5,000 genes, like, or 3,000 genes. So basically each cell you have more than 3,000 or 5,000 numbers that describe the cell. But when we visualize the data on the screen, like, we are essentially representing each cell with just X and Y coordinates. So all the 5,000 numbers became just 2 numbers. How do we do this while still retaining any useful information? We still see the distribution of cell type, and when we can still see batch effect or the remove batch effect from the visualization. So there must be some clever way that we can reduce 5,000 numbers down to just 2 to visualize on the screen. So this is the first technique that we want to talk about. So in our data, like in transcriptomic data, we think about the dimension of the data, for example, maybe with the number of genes. If we measure expression level of 4 genes, we think that our data has 4 dimensions. If we measure the expression level of 10,000 genes, maybe our data has 10,000 dimensions. But in biology, like we know that even though we measure the expression level for all genes, we do so because we don't know which gene will be relevant in our analysis. So we are actually measuring genes that are not changed by the treatment or genes that are unaffected by the disease. So in our data, like even though we have measurement from 10,000 genes, only some of them are actually relevant in the biological systems or treatments or disease that we are interested in. So the true dimension of the data might not be the actual number of genes. It might be much less, depending on the complexity of your biological system. Just to illustrate, if we have two data set or 2 conditions, representing by a different cube, like one with a blue cube and the other one with a red cube. The goal of the visualization is we want to distinguish. We want to be able to show that we have two groups of data in our data set. So basically, we don't really want to see the whole cube. Like we just want to say that there are 2 cubes, there are 2 objects. And if we want to just show that there are 2 objects in our data, we don't need all 3 dimensions. We can just do a cross section of the cube, right? And we have 2 dimensions. And we can still see that there are 2 groups of data in this data set. We can even reduce the square into just a line, and we can still see that there are two groups of data in our data set. So the concept of dimensionality reduction, which is basically to reduce the dimension, is to do so while still preserving or maintaining the key information that we want to convey. So in the context of transcriptomics, we have 10,000 genes, but maybe our disease or our treatment only affect one pathway, which might involve maybe 10 genes or 12 genes. So the idea of dimensional reduction is, can we represent the change that are affected by the treatment or the disease by using just one or two numbers, just few numbers. Okay, to give you more illustration of how this is done, so let's look at one example. So we have a data set consisting of image. So this image are handwriting. So we ask people to write number from 0 to 9, and we take the image and then we convert it into files. There are two data sets, USPS and MNIST. So these are collected separately from two different populations. And they might have a different pens, like different criteria of collecting the data. You can already notice that in the USPS images, the numbers are very thick, the line is very thick. But for the MNIST, the line is very thin. So there's already some systematic, some batch effect between these two data sets. So when you visualize the data, let's see onto the screen, like onto 2 dimensions. Even with we're going to go over some of these techniques, but even with the simple techniques, you can already see that the data from USPS and data from MNIST are clearly separated. So if you color the data point based on the data source, you can already see a good separation between the two. But if you color the data point using the numbers from 0 to 9, some are very clearly separated. In the orange here, like you can see that it's very clearly separated, but the other numbers are still mixed. And if you check like what these oranges data points are, so it's the second group. So it starts from 0. So the second group is number 1. And the reason why number 1 is clearly distinct is like, if you compare just by eye, like, number 1 is the only number that has no curve at all. So even with no other information, dimensionality reduction techniques can recognize the fact that number 1 has a different distribution of pixels or characteristics compared to other numbers. So it can already separate out this group, even without being told like what the numbers are. But if you use like a more advanced technique, and we will see this with single cell data, when you feed just the image of the numbers, and we don't tell the model which data source the image is from and which number is contained in the image. But with dimensionality reduction technique, we can already separate out not just the data source, but also individual numbers. So with powerful techniques, we can group data without having to know what type or what class they are from. So let's go over some of these techniques because you will see them in single cells literature and probably in your work as well. So the first technique is called principal component analysis, or PCA. Some of you might have seen this maybe from statistics class or mathematics class before. So principal component analysis, if we want to talk about what's the key idea behind this technique. The key idea is very simple. When you have a data set and you have no information about how many cell types are in the data set at all, like you have no information at all, the only thing that can guide you is how the data are distributed, the variance of the data. And the assumption behind PCA is that the direction with the highest variance, like the direction where the data points are most scattered, this is the direction that have the most information because it can separate out your data set. So if you look at this kind of toy example, we have 3 maybe cell type, and each cell type we have 3 cells each. When we want to visualize these cells, like 9 cells, onto the screen, and we have to put them on the screen. That means we have to reduce the dimension to maybe just the y-axis or just the x-axis, or maybe with some diagonal line. The idea behind PCA is if you compare these three types of projection, like we want to reduce from two dimension x and y to just one number. So we have a choice. We can remove x. Everything becomes just the y coordinate. We can remove the y coordinate. Everything becomes just the x coordinate. Or maybe we come up with a combination of x and y. Maybe x plus y, for example. And the idea is like this projection to the y-axis, we lose the x coordinate. It's bad because now these two cell types are indistinguishable from each other. If we project onto the x-axis, these two cell types are now indistinguishable from each other. So these two projections lose information. So the idea of PCA is, so let's try to find a direction where if you project all the 9 data points, they are more separated, like the variance is the highest. And in this case, this diagonal direction is where the variance is the highest, because all the 9 data points are all spread out. In the other direction, you have 6 data points being come together and 3 data points come together. But in this diagonal direction, you have all 9 data points being spread out. So this is the direction of maximal variance. So if you imagine that your data consists of multiple cell types, if you want to project the data or visualize the data in such a way that you see the cell type being separated, that means you are trying to create maximum variance on your screen. You want the data point from all the cell types to be separated. You want high variance. So that's why PCA is a good technique. It's a good initial technique to visualize the data, because most of the time the direction with the highest variance can give you a lot of information. So the procedure is this. If you have a new data set which looks like this, we have 3 measurements, but we can see by eye that the direction with the highest variance is probably the diagonal. So what PCA does is it's trying to fit this kind of ellipsoid shape, like a rugby shape, into your data. And then the major axis will be your first direction. This is the first axis that has the highest variance. The second one will be the next axis that have less variance, and then the third one that have even less variance. And one interpretation, like why this operation is useful, is because when we make the measurement, like as a biologist, when we make the measurement about the cells or about the animal or about the plant, what we can measure is limited by the technology. We can, for example, we can measure the drive rate, we can measure the gene expression, but in sometimes, like what's the factor that can explain the disease, like is multidimensional. It requires multiple genes combined together to create like markers. But because our measurement technology measure expression for one gene at a time, so the axis that we have in the original data is limited by the technology. We cannot measure the diagonal direction because we have no assay for that. We have no technology for that. So PCA, basically what it does, it transforms the data from what you can measure into the direction that are useful for making prediction or for understanding the biology, which we cannot measure directly. So the idea of PCA basically is to change your original axis into a new set of axes that are sorted by variance. So the original axis might not be sorted by anything, right? But the new axis will be sorted by variance. So when you see in several papers, when they try to visualize like, how much variance that the PCA can capture in each dimension, in each axis, you will see this kind of bar plot that the numbers go from high to low. So the first bar here will be the amount of variance that can be captured by the first axis, the major axis. So in this case, this means that 15% of the variance, if you consider that your data is 100%, 15% of the distribution of the variance can be explained by the first direction. And then an additional 10% can be explained by the second dimension. Another maybe 9% can be captured by the third dimension, the third axis. and then another 7.5% can be captured by the fourth axis. So by default, PCA doesn't reduce dimension. If your starting data has 3 dimensions, you will have a 3 axis out from PCA because it just kind of restructure your dimension, your axis. It doesn't reduce it. But because PCA does so by sorting from high variance to low variance, so what people usually does is to plot the cumulative variance. So the first axis only capture 15%. The first and the second together capture 25%. The first, the second, and third together capture 35%. So if we add more and more dimension, eventually you can capture maybe 80% or 90% of the information in your original data. And at certain point, you can say, okay, I think 10% of my data may be noise, might be technical variance, maybe biological variance. I don't too care about those. So instead of using the whole maybe 38 genes, we can stop at 18 genes, and we can still create a new axis from PCA that can explain 90% of our transcriptomics data. So one benefit of PCA is when PCA creates these axes, it has meaning. So I don't think you learn linear algebra to explain simply. Like when we visualize the data using PCA, let's say this is the first axis from the PCA, and the Y here is the second axis from the PCA. And we see this kind of distribution. We see maybe patient data or maybe single cell data, and we see that two different groups, like normal cells and different cells, or normal patient or different patient, are distributed according to the x-axis, according to the first axis from the PCA. So we want to make interpretation, right? So what measurement of which gene can explain why these cells or these patients are located on the right of the graph, the orange data point. So in PCA, if you want to understand each axis in PCA is a linear combination of all of your measurements. So for example, if you have a transcriptomic data with 100 genes, your first principal component, your first axis from the PCA, is going to be some coefficient times the expression of gene 1 plus some coefficient times the expression of gene 2 and plus some coefficient times the expression of gene 3. So if you are focusing on the first dimension, you can ask what are the coefficients for all the genes along this direction. And because we are interested in the orange data point that are located to the right, so these are data points with high coordinates, high score on the first PCA axis. So we look for genes or look for input variable with high positive coefficient. So whatever this measurement is, whatever this gene is, these are the genes that explain why these cells or these patients have high score. And for genes or input variable with negative coefficients, these are the ones that are correlated with being normal from the normal group. So good thing about PCA is when you visualize and you see some direction that is interesting, you can go back into the model and ask which coefficient, which gene or which input variable explain that direction. You can ask, you can get one coefficient like this for the first axis. You can get a different set of coefficient for the second axis. So you can interpret each direction. So let me show you another example. So in this case, I take data from multiple batch. So omics data from multiple batch, for example. And I visualize the data using PCA. And I ask maybe for 3 different genes. I want to see the expression level or maybe other score for the gene. So I can see that for gene number 1 is correlated with the first dimension of the PCA, first axis. Cells with high expression of gene 1 are located to the right of the graph. Genes with high expression for cells with high expression for gene 2 are located to the left. So it's the opposite from gene number 1. So gene 1 and gene 2 might tell a different story. And for gene number 3, now it's ordered by the y-axis. So this is based on the principal component number 2. So this also gene number 3 convey a different information than gene number 1 and number 2. And when I color the data point by the batch ID, so these are from different hospitals, for example, you don't see any pattern. So this is a good sign that there's no batch effect, because if there is batch effect, cells or patients from the same experiment or from the same data source will be clustered together somewhere. So we can use PCA to check for batch effect as well, okay, by visualizing. Okay, so just to summarize, so PCA is easy to use because it assumes that the index of the highest variance is where we want to visualize the data. It doesn't do anything complicated. And the way that it tries to find the dimension, this axis is a linear combination of the original input, so you can interpret it. So PCA is interpretable. You can understand why it places a certain cell in a certain position. But one thing that's kind of, this might be technical, but one thing that is a limitation of PCA is the distance, like the way that it places individual cells or patient or sample, is based on what's called Euclidean distance, which is just a straight line between two data points. So PCA only works if your data, like when you compare the similarity between your data, like between two data points, you want to use Euclidean distance. But in biology, we have many other ways to tell if two cells are similar, if two patients are similar. That is not Euclidean. So PCA can be limited in some cases. So some of you might have heard about principal coordinate analysis before, PCoA. This is what people in biology use when their data is not described by Euclidean distance. For example, if you study microbiome, you want to compare two communities of microbiome. They have different composition. In our metagenomics lecture, we learn about uniform distance, we learn about Bray-Curtis distance. Those are not Euclidean. So in biology, different data type require different definition of similarity. That's not Euclidean. So just to show you some more variety of distance. So Euclidean is a distance that we are used to in the real world. When we see two data points being far or being near each other in 2D, 3D, these are all Euclidean. Our eyes interpret distance as Euclidean. But sometimes when we care about two data points, we care about the location, the angular location. Like on the clock, if you want to tell if two times are similar, you look at the angle between the needles. You don't look at the distance, you look at the angle. So sometimes we need to calculate the angle instead of the actual distance. If we are considering like mutation data, when we compare two sequences, two DNA or two protein sequence, we look at the position where they differ. So this is called Hamming distance or edit distance. The number of mutations that has to occur to change A to B. This is not Euclidean. If you have compared two sets before, actually when we talk about functional enrichment, it's a comparison between two sets. Like one set is a set of differentially expressed genes. The other set is a set of genes with certain function or from certain pathways. We want to compare the overlap, like the intersection between two sets. Sometimes this is called a Jacquard distance, also not Euclidean. So in biology, we have many things that don't fit the Euclidean. So people have to come up with a different technique that allow us to use any kind of distance and it still work. So let's look at the framework. So the first terminology that I want to introduce is called the pairwise distance matrix. And as the name implies, so this is going to be a pairwise. So for every pair of sample that you have in your data, if you have A, B, C, D, E, F, G example, you're going to pair up every possibility of them. And for each pair, you're going to calculate the distance, which doesn't have to be Euclidean. It can be correlation in gene expression, can be the Bray-Curtis distance in microbial abundance, it can be the intersect between two sets. But you want to calculate the distance between every pair of samples. We're going to use this to help the dimensionality reduction process. And the idea is, like, if you have sample A and sample D, like in this example, A and D are near each other, like distance only 8. B and F are also very near each other, like very similar to each other, with a distance of 1, so they're close by. They are similar. But A and B, like 19, A and C, 27, B and C, 31. So these are not similar, these are different. If we want to visualize like this data set onto the screen, what we can probably agree upon is we want to place B and F next to each other because they are really similar. We want to see them next to each other on the screen. We might want to see A and D also near each other on the screen because they are similar. But A and B, like, we want to see them further apart. Like, we want to know that they are not from the same group, probably not from the same cell type, for example. So we have some criteria. Like a good visualization or a good dimensionality reduction result should preserve like, the similarity between our samples to be the same or similar to this kind of pairwise distance matrix. So the idea, and this idea will apply to every dimensionality reduction technique that you see in single cell analysis. And the idea is everyone starts with the same thing. We have the measurement data. This is experimental data. So we have samples, and we might have expression level for thousands of G, and for 10,000 G. What we can do to the data is we can calculate the similarity between cells, the similarity between samples, using any kind of distance that you think is appropriate. What we want to get at the end, let's say if our target is the visualization of single cell data, we want to see this data point on the screen, maybe in 2 dimensions. So let's say someone, like some kind of God, tell you that this is the answer on the screen. This will capture the characteristic of your data. If you know this, like, let's assume that if you know this, like, if someone tells you that this is a good idea, what you can also do is take the coordinates that the samples are put on the screen and calculate the Euclidean distance between them, because these are the distance that our eye is seeing on the screen. If the position of the cells or the sample on the screen, if this is a good arrangement of the samples, these 2 metrics should be similar. Samples that are similar to each other should be placed near each other on the screen. Samples that are not similar, that samples that are different from each other, should be placed far away on the screen. So the idea of our dimensionality reduction technique essentially is to find position on the screen, so for simplicity, the location on the screen in such a way that when we calculate back the distance on the screen that we see is somehow similar or correlated with the distance that you calculate from your original data. If an algorithm can do this, like, you get a good visualization of the data that preserves like whether the samples are near or far from each other, reflecting how similar they are. But the process, like whether you use correlation, maybe Pearson correlation, Spearman correlation, and the algorithm to find the position of the cells or the sample on the screen, these differ from technique to technique, but the ideas are all the same. They want to optimize the similarity between distance that your eye can see versus the similarity that you calculate from your data. We are not going to go into like how they actually calculate it, and that might be too difficult. But the idea, at least this idea, should give you inspiration that this is a process that is natural and can be done. So, just to give you slightly more complex situation, when we analyze single cell data, we have data from different cell types. And we know from biology that some cell types might have a diverse gene expression profile. The gene expression can change a lot, but it's still the same cell type, the same phenotype. But some cell type, the expression level might be tightly regulated. So when you visualize the data, you can, if you don't do anything to the data, you're going to see that some cell type, they will be tightly clustered because their expression profiles are tightly regulated. But some cell types can be everywhere. The genes can be up and down, but it's still the same cell. It can still maintain the same function. One of the limitations of the techniques that we mentioned all the way up to here is you have to pick one definition of similarity, and you have to apply it to every cell. For example, if you measure a distance of d, imagine d is just some number. This distance, whether you interpret it as cells being similar or different from each other, you have to apply this criteria to the whole data. And based on this the different invariance across cell type, you can tell that this distance of d, if you apply it to the blue cell type, the two cells are not similar at all. Distance of d is very far in the context of the blue cell type. But in the context of the red cell type, if you pick two cells and their difference are d, they're still the same cell type. So the same distance can mean different things depending on the cell type, depending on the variability in the gene expression. So these are the severe limitations that other techniques have that cannot be applied to single cell data. So nowadays, when you look at papers on single cell, the technique that I'm using most of the time are these two. So these are the new generation of techniques, tSNE and UMAP. So the name might sound very scary, but the idea is similar. Okay, so let's look at the idea. So what they want to solve is our data can have different variants, or to put it another way, our cell type can have different density. Some cell type is very dense, some cell type is more sparse. Can we handle this in doing the dimensionality reduction steps? So first, these two techniques, what they do is they have to be able to recognize the density of the data. By eye, it's very easy. You can look at these two situations and you can tell that the left-hand side is dense, the right-hand side is sparse. But as a computational biologist, if we want to do computation, we cannot use our eye. We have to translate this problem into some algorithms that the computer can handle. So how to measure data density? There are two ways. A naive approach, if we want to measure density, by definition, density is the number of data points within a certain area. That's the definition of density. So you might count the number of data points within a certain distance. You draw a fixed distance, maybe of 5, a distance of 5, and then you count how many data points are in this area. And you count 1, 2, 3, 4, 5. So that's 5 data points. The problem is, how do you know which distance to pick when you change from dataset to dataset? Because one dataset, distance of 5, you might find many cells. In a different dataset, distance of 5, you might not find anything. So if you want to count the density, if you specify the distance, you have to keep changing it from dataset to dataset. The other way to do this, cleverly, is to measure the distance to the nearest neighbor, maybe the fourth nearest neighbor in this example. So whatever the fourth nearest neighbor is, how far it is, we're going to find. So let's say the yellow point is our starting point. We want to identify the nearest cells to this yellow cell. So the nearest one might be this, the second nearest this, the third nearest this one, and the fourth nearest is the blue one. And then we measure the distance between yellow and blue. So we get one measurement. In the second situation, we also check the nearest cells is this one, the second one, the third one, and then the fourth one. So the first one I highlight with blue. So once we find the fourth nearest cells, we calculate the distance. So that if the distance to the fourth nearest cells is small, this tells us that the density must be high, right? Because you already find 4 cells within this short distance. But in the second situation, the distance to the fourth cell is very far. So this means your density is low, because you have to go very far to find the fourth cells in the neighborhood. So instead of defining the distance as a unit, right, we define the fourth nearest neighbor, maybe the 10th nearest neighbor, maybe the 20th nearest neighbor, which works every time. Right? And we measure the distance from the center of the cells to that neighbor, and that distance will tell us whether the density is high or low. So once we determine the density based on this distance, we can interpret other cells easily. Let's say we want to interpret whether cells in red and the cells in yellow are similar or not. We can compare the distance d, like, which are equal, like this. In the 2 examples, the distance from yellow to red are both d, so they are the same. But when we interpret in this situation, we can interpret this distance d relative to the distance between yellow and blue. In the first example, the distance between yellow and blue is less than d. So this means this red cell is not similar to the yellow cell. But in the second situation, this distance of d is even shorter than the distance between yellow and blue. So this means the red cells here is similar to the yellow cell. So we can interpret that by comparing to this measure of density. So what these techniques actually does is it tries to calculate, to estimate the property that 2 data points, red and yellow, are they going to be neighbors with each other? And by being neighbors means that they are near each other. And when we visualize the data on the screen, if they are neighbors, they're going to be placed near each other on the screen. So in this case, they are going to, so this is from the normal distribution, but if you don't remember that, it's okay. What we want to focus is, we see the distance d here. This is the distance d in red between the cells that we are interested in. We normalize this distance by the sigma in green, and the sigma in green is the density of the data point around that area. So we normalize the distance by the sigma. So we can take care of the difference in density. And not only that, if we want to say that cells in red and the cells in yellow are near each other, are similar to each other, the cells in red have to compete with other cells nearby. If the cells in red are more similar to cells in yellow compared to other neighbors, then they are neighbors. But if the cells in red are further away compared to other nearby cells, the red cell is considered not a neighbor. So in this formula, the numerator normalizes the distance with the density. The denominator, I forgot the term, but sorry. So the bottom part, this normalized also by the distance between yellow cells and nearby cells. So it's a double normalization. First by the density, and also by comparing red cells to other cells nearby. So these two together give a measure of whether yellow and red are they similar or not by these two factors. And from this calculation, they're going to follow the same framework that we saw earlier to project from the single cell data. From single cell data, they compute the property of being neighbors using the formula that we saw earlier. And at the end, there's one thing that differs between the previous framework and this one. And that is, when we want to show the data on the screen, we want to make the cells are nice and separated. So we want to control the density on the screen. So in the original data, some cell might be sparse, some cell might be dense. But when we visualize on the screen, we want the image to look nice. So what tSNE does, what this technique does is, when it projects the data onto the screen. When it calculates the distance from this side, from the visualization side, it fixes all the sigma, the density, to be 1 throughout the dataset. So the original data can have different density depending on cell type, but the visualization, what you are going to see after this, the density is going to be fixed as 1 all over the data. So you're going to have a nice visualization. So it can solve 2 problems in one go, right? Both to reduce the dimension of the data and also to make the visualization easy for human eye to interpret. Okay. So when you use this technique, you're going to get to use these techniques. One of the parameters that they are going to ask you, one of the key parameters is when we want to estimate the density, right? And we say that we can estimate the density by measuring the distance to the k nearest neighbor. In our example, we use k to be 4. But depending on the data set, this number, sometimes you have to change. So sometimes you might use 5, like the 5 nearest neighbor. Sometimes you have to increase to 10 nearest neighbor. Sometimes 25, sometimes 50. The criteria for selecting the good k, like the number k here, is there's only one issue. If your k is too small, let's say you're looking at the 5 nearest neighbor, and you have some cells that are outliers, but they are similar to each other. If you have more than 5 outliers, if you only look at the 5 nearest neighbor, those 5 outliers are going to cluster with each other and not with anything else, because they are looking among themselves, like they are all the k nearest, they are all the 5 nearest neighbor of each other. So they're going to form small clusters when your k is too small. But when you increase the k further, you can see that there's almost no harm in being too large. Like k from 10, 25, and 50. These 3 settings give you almost the same interpretation. You see the blue cells being clearly separated. You see the red cells and the green cells separated but still near each other. So you have the same interpretation with multiple settings. So the recommendation from the developer of the techniques is that you should try different value of the k. In that program, they will call this perplexity. But essentially, it's the number of neighbors to calculate to determine the density. You can try changing the k, and what you want to see is some patterns or some grouping of the cells that are consistent as you change k. You don't want something that unique to k to equal to 5. You want something that is the same regardless of the k that you set. And this can help you to, this can guide you to select the appropriate k, which there are a lot of. Everything in this range works. So you have to worry about being like trying k equal to 11, 12, 13, 14. Everything in the same range will work. Okay, so let's see how tSNE and a related technique, UMAP, perform on single cell data. So from here, there are multiple cell types, color with different colors. And when we visualize by maybe focusing on certain genes, so we can color, when we visualize single cell data, we can either color by the cell type, if we know the cell type, or we can color by the gene expression. And in this case, you're going to see that both techniques can cluster the same cell type together. All the green cell types are next to each other, connect to each other. The blue cell types are connected to each other. The red cell types are connected to each other. tSNE also, maybe to a lesser extent than UMAP. When you look at the gene expression, you're going to see that they are clustering, they are putting the cell with similar expression next to each other. So cells with high expression of this gene are being placed near each other. But if they come from different cell types, even though this gene are highly expressed, the other gene might be different. So you can see multiple locations with high expression of a gene if there are multiple cell types that express the same gene. So if you compare this technique from this visualization, you can see that tSNE, most of the clusters, most of the cell types are quite rounded. compared to UMAP, you can have rounded cluster, you can have a more scattered cluster, you can have like a path between multiple clusters. So these two techniques operate on slightly different assumptions. So tSNE, the goal of the technique is to show you the clusters of the cell. So you're going to get all the cells being clumped in kind of rounded clusters. So it's easy to see the clusters. But UMAP, they also assume that when you have multiple cell types, some of them may be related. One cell type might be able to differentiate into another cell type. So in UMAP, they want to outline the path that connect multiple cell types as well. So in that techniques, they are able to kind of lay out the cells such that you can see the transition from one to another more clearly than in tSNE. Because in tSNE, even though the cell types are next to each other, but it's difficult to see like where they are connected. They are all rounded. Another example that shows this more clearly. So this is a immune data set. You have the multipotent progenitor that can differentiate it into different immune cell types. With tSNE, you get a good separation of the cell type, and you see multipotent progenitor like in the middle. But it's still unclear like how cells differentiated into each cell type based on the arrangement from tSNE. But with UMAP, what people found when they first developed UMAP is that you can see the location that goes from multiple potent progenitor to different cell types, all separated out into different directions. So it's easier to see cell development with UMAP. So depending on your task, like if you have cell types that are not related, like all differentiated, tSNE can help you identify the cell type, maybe easier. But if you have like a stem cell development data, like you want to see how cells change from one type to another, usually UMAP can give you a better idea of the trajectory. Okay. So now we have seen the techniques that can allow us to summarize single cell transcriptomic data from thousands of measurements for each cell down to just X and Y coordinates. And we can still see meaningful biological information on the screen. So after we can visualize, what next can we do? There are two main goals in single cell analysis. First is to identify the cell type, like to cluster the cell type. And the next is to identify development trajectory from one cell type to another. So to show as a diagram, we start with single cell data where we have no idea about the type of individual cells. We want to do dimensionality reduction to visualize the data on the screen. And then we want to perform clustering. We want to group cell types, cells from the same type together so that we can color them and see like, if there are cell types that are separate from each other, or are there cell types that might develop from each other, like they are next to each other. And the next step would be trajectory reconstruction. We want to understand how cells develop from one type to another. And once we are able to learn this trajectory, we can look at the expression of genes along the trajectory. And we can identify genes that are upregulated or downregulated as cells change type. So we understand the switch that we can turn on or off if we want to direct our cells to change. So we can learn that from single cell data. Okay, just to give you one example from research. So in this study, they want to understand how embryonic stem cells differentiated into multiple cell types. So first, we get the data and then we visualize it on the screen. So we got the visualization. Next, the author cluster cells with similar expression profiles to create the coloring of cells with the same type, from embryonic stem cells in red to differentiated cells in blue. So once we have this visualization, then we want to infer the trajectory. We already know the embryonic stem cells have to be the start. We have to infer how cells develop, flow into multiple cell types. We want to infer this arrow. And then, once we know how cells change from one type to another, we can infer all the genes that are the switches that are required for the cell to change from one stage to another. And this can benefit people who study stem cells or people who want to differentiate cells through certain tasks. So from this point, we're going to talk about technique for these two analysis, how we infer cell type and how we infer the trajectory. So to infer cell type, the first step, we want to be able to cluster cells. We want to group cells with similar expression together. We'll talk more about this in the biological network lecture, but to give you a sense of what it's doing. So we have visualization of the cells using dimensionality reduction technique. What we can do is we can connect cells with similar gene expression. We might have a threshold that, okay, if the correlation in gene expression is above 0.7, then we draw a line, we draw an edge. So we have some connection between cells with similar gene expression, connection between cells with similar expression. What we want to do, which is called network clustering, we want to group cells, we want to color the cells, so that cells with the same color, they are highly connected to each other. But cells with different color, they are not connected. And we can infer that these are different cell types. They are similar to each other within the same type, and they are dissimilar or different from each other across time. So what are the criteria that we can use to look at this graph, look at this network, right? And then define that, this is my cluster, this is my cluster. One thing that we can calculate, we can think about what is a good clustering result. If we compare these two clustering, we know that the top one looks good. Right? We have all the blue colors connect to each other, all the red connect to each other. Compared to the bottom one, we have blue connected to red everywhere. So one of the criteria that we can use to cluster cells with each other, once we draw the line between cells that are similar, is called a modularity score. And this basically calculates the ratio between number of edges within the same color compared to the edges between different colors. We want to maximize this score. We want more edges between within clusters and less edges across clusters. And some of the algorithms that we will talk in later lecture are called Rowan and Redden. So these are network clustering techniques that people use on single cell data to identify which cells are from the same type. But at this point, we still don't know what type they are. We only know that they are similar. They are probably from the same cell type, but we don't know what type. So the next step would be, how can we define the biological cell type onto the data? There are essentially two ways. First, you can compare your data to other people's single cell data of the same tissue. And if they already perform annotation, you can compare the similarity in your cells, expression pattern, to that study. And you can infer the same cell type from that study. Another technique is you can, if you want to identify known cell type, so usually known cell type, people will have some marker genes for them. Genes that are known to be highly expressed in those cells. You can visualize your data using the expression for those genes. And then if you can identify some clusters that show high expression for those genes, you can classify them as that type. So as shown in this example. So we are focusing on these clusters. And let's say we know that these must be some type of macrophage. So we take the CD68, which is one of the markers for macrophage. And it's high, only in this cluster. So we have a good confidence that this cluster of cells probably is a macrophage. But one thing that I want you to be always careful of is when we cluster the cells, we have the same cells next to each other, but sometimes biology can be more complicated. So within macrophage, there are also multiple types of macrophage, inflammatory or non-inflammatory. So sometimes you run into a situation where the cell type, like there can be sub-cell type, and they can have individual markers. So you should try. So in this case, people know that for inflammatory macrophage, the LYC gene could be high. And for non-inflammatory, macro gene could be high. So in this case, you can see that in the cluster for macrophage, the visualization algorithm, tSNE, is already smart enough to know that among macrophage, which have CD68 expression, some of them has high LYC expression, and they are grouped toward the top. And some of them have high macro expression, and they are grouped toward the bottom. So in this case, even though you might see a single cluster, but when you visualize with known genes, known markers, you might identify that, oh wait, actually I have more than one cell type next to each other. So you should do a lot of literature review to identify marker genes and visualize them on your data to see what the cell clusters mean. So this is very common to see multiple cell types next to each other because they share some markers and they have some unique markers of their own. Okay. So once we can make sense of the cell type in the data, now we want to know if we can infer how cells develop from one type to another. So this is the trajectory analysis. Sometimes people will call this a pseudotime analysis. And that's because usually when you want to analyze cell development, usually you have to culture the cells and get one sample from day 0, and then maybe get another sample from day 1, another sample from day 6, another sample from day 12, so that you can see the development over time. But in single cells, because we know that each cells are in different stages, so they can actually show us the development pathways. But they are not true developmental datasets because you are not starting from the same cell. In time series experiment, you start from the same batch and you culture them for longer and longer time. But for single cells, you capture the sample only once and you assume that they represent cells from different age, different stage. It's not a real time, it's a pseudo representation of time. So people call it pseudo-time analysis. Okay, so our hope is to be able to draw this kind of arrow that goes from one cell type to another. Okay, so there are many ways that people came up with for this purpose. So computer science people say that, okay, if we assume that cells develop from one type to another, why don't we assume that cells go from this stage or this type to the next stage or the next type along the shortest route? If we assume that biology takes the simplest option, if we can identify this part in black that connect as few cells as possible, and these cells must be similar. Cells that are connected must still be similar, have similar gene expression profile. But we want to identify the connectivity among these cells that are shortest and can take us all the way from this cell to this cell. If we can do a computer science algorithm that finds the shortest path, this might be one possible answer. So this is the first idea that people came up with. Just assume that nature follows the shortest possible route. But in real life, we know that it's not that case. Cells can have very variable gene expression. There is a lot of probability involved that how do you change from one to another. So people from physics propose another idea. If we assume that cells, the expression level inside the cells can change in a continuous pattern. So you can think of this as cells change from one expression to another gradually, as particle, things like a physicist, as a particle that moves in space. The particle moves from one location to another with random probability, like a diffusion model, diffusion process, or random walk process. So what they are doing is they are calculating the cell-cell similarity to create which cells can move to each other. If they have similar gene expression, they probably can change into each other. Then we calculate the probability that cells will go from one gene expression profile to another gene expression profile to another profile, following something like diffusion process, like molecules, particles moving in space. And then we will have some kind of a probability that if you start from this position and your cells can change expression gradually, what's the chance that it will eventually arrive at another location? That will be your probability of starting from one cell type and then reaching another. If the probability is high, that means the cells can go from one type to another along that route. If the probability is low, that means the cells probably doesn't evolve or develop that way. So they infer the path from that probability. So this is like the second generation of techniques. We infer the likelihood that cells can change expression level from one stage to the next, and then draw the graph that connect paths with high probability. And we can understand how cells change from one to another. But one of the limitations of these two techniques is, even if we can connect a cell with similar gene expression, how do we know if A develops into B or B develops into A? Similarity doesn't give direction. So all these techniques, they ask you as a biologist to tell them which is the starting point for cell development and what's the end point. So you have to define the start and the end. And the algorithm will identify the shortest route or identify the most likely probability, the most likely route for you. But what if we don't want to do that? Right? What if we don't know which cell type is the start and which cell type is the end? We want to infer from the data automatically. Can we do that? So the third generation of techniques, one of them is called RNA velocity. And when we mention velocity, like velocity is a vector, so it has a direction. So what this technique implies is from your transcriptomic data, they're going to be able to tell you whether cells A differentiate into B or B differentiate into A. They want to give you the direction. And one key assumption that they are going to use for this technique is based on unspliced and spliced isoform. So up to this point, like, we never care about unspliced or spliced RNA. We just count the number of reads and we get the gene expression. But if we think about the mechanisms that occur inside the cells, right? If we want to activate a gene, the gene must first be transcribed into an unspliced form. And then the spliced molecules will be spliced by the machinery to get and then be transferred into protein. So if you plot the graph in your cells when a gene is turned on, you're going to see an increase in unspliced RNA first, followed by a delay increase in spliced isoform. So that means that if you can measure the ratio between unspliced isoform and spliced isoform, if you can measure this ratio, if it's high, that tells you that the cell just started activating the gene. If the ratio is small, that means the gene has already been activated a long time ago. So based on the ratio between unspliced and spliced transcript, you can tell the timing of gene activation. You can tell whether the cell just changed or the cell has changed for a while. So RNA velocity model use this ratio to order the cells. If you see high ratio, cell just start activating some gene. If the ratio is low, the cell is probably after that, after the activation. So this gives you the direction just from the data without you having to tell anything. One key assumption though, we are assuming that activation of a gene means the direction of development. Some of you might think that some development might depend on repression, like not activation. But we can assume that most of the time, like, activation should be the preferred method. Because if you have to develop after replacing something, like, that means the cells have to constantly express that gene in order to be replaced when it wants to differentiate. that require energy. But activation, that means the gene doesn't do anything to that, the cells doesn't do anything to that gene until it is needed. So people use this assumption to believe that activation is a sign of development. We want to activate something when it's needed, and that's to go forward in the development. So let's see, like this is a nice idea, but can we trust it completely? Like, it sounds nice. So when people first develop this model, they want to prove that this works. So if we want to prove, to show that the cells develop as the gene activates, what kind of genes are good for this? One class of gene is the circadian genes. So circadian genes are genes whose expression cycle throughout the day as we live. So this study looked at the ratio of unspliced and spliced RNA for circadian gene all throughout the day. And they found that if you draw the arrow based on the time of the day, like midnight, 3 a.m., 6 a.m., 9 a.m., noon, 3 p.m., 6 p.m., 9 p.m., for any gene in the circadian rhythm, you see this kind of cycle, depending on when it's activated. So FGF1 might be activated at 6 a.m., and the ratio of unspliced goes up from there, like at 9 a.m., at noon, and then it goes down. So this means that it's already on the way down. It's activated in the morning, and then at night this gene is downregulated. For CBS, this is activated in the afternoon, and then early morning it goes down. It went down. So this data set can prove the concept that by following the direction of activation of the gene, we can follow the development in time. And this doesn't require you to change anything in your single cell RNA sequencing data. The newer techniques, they are all full-length sequencing techniques. So you need to see the entire transcriptome, the entire length of the isoform. And from that, you can see the intron. If the read has an intron, then that means it's unspliced. If the read contains the spliced junction, that already tells you it's spliced. So this ratio can be readily calculated from existing single-cell RNA-seq data. So up to this point, like we have seen the method to visualize single-cell data, how we can identify cell type in the data, how we can infer the trajectory in the data. One of the things that I highlight at the start, and I haven't mentioned yet, is the choice of genes that we are going to analyze have an impact on all the results. This is important because in every pipeline that you are going to encounter, there's going to be one set that mentions this term, highly variable genes. And the idea is, like, let's imagine you perform single-cell RNA-seq, you get transcriptome data for 5,000 genes. Most of those genes are not differentially expressed. Most of the genes are similar for all cells. Only some of the genes can distinguish cell types. So what happens in your data is, when you want to visualize the data, when you want to cluster the data, one of the things that we want to calculate is the similarity between cells. We talked about one factor that affects the similarity calculation, and that is like which metric or which distance function you want to use. Like Euclidean, correlation, Bray-Curtis, and others, and so on. Those definitions of distance directly affect the similarity between cells that you are going to get, the numbers that you are getting. But the second factor that's rarely discussed is the gene set that you are using to calculate. So imagine like you have 5,000 cells, you have 5,000 genes. Maybe only 100 genes are unique to different cell types. So you have 4,900 genes that are not telling you anything about the cell type. If you calculate the similarity using all 5,000 genes, that means 4,500 out of 5,000 is giving you noises. Only 100 genes give you the signal across cell type. So in many analyses, they're going to ask you not to use all genes because that will dilute the signal. They're going to ask you to use highly variable genes with the assumption that if you have different cell types, like genes that are unique to cell type is going to be high in some cell type, low in some cell type. So it's going to be highly variable. But genes that are not a signature or not a marker for any cell type, the expression level will be the same across. So by looking at the variance of the genes, we can gauge how useful it is for our analysis. But it's not only that. If your data set only contains one tissue, and your only source of variability is the cell type, using high-level variable gene is good enough. But if your data set contains not only just different cell type, but you combine data from different treatment, like untreated or treated, or treatment with multiple concentration, like multiple dosage. And let's say you also include data from multiple donors, like to look for biological effect. In your genes, like in your 5,000 genes, some will be useful for distinguishing cell type, some will be differentially expressed across conditions, and some might be differentially expressed across donors. So if you pick genes in your analysis, if you pick genes that are highly expressed, but their variation actually came from treatment, because the treatment effect might be stronger than cell type, then your visualization is going to distinguish treatment and not cell type, because the choice of gene are the genes that are differentially expressed across conditions. If you use genes that are differentially expressed across donors, your visualization and your analysis, you're going to see cells from different donors being separated. And that's not what we want. When we analyze single cell data, our goal usually is to understand different cell types. So we want to identify highly variable genes that are useful for distinguishing cell type. So in the software that you will run into in the future, when they ask you to use highly variable gene, they're going to ask you like how to calculate the variance for this gene. So if you select within sample, so this means that you are controlling for treatment and donor. For each sample, it's only single donor, single treatment condition. So if you calculate the variance of a gene within sample, that captures only the cell type. So this is a recommended setting. You want highly variable gene within sample, not across sample, because across sample you will get genes that are different across treatment and different across donor. You want within sample highly variable gene. So that's one of the key. If in the assignment, I'm still deciding, but I may ask you to try a different highly variable gene settings and see how you can see different information in the visualization. Okay, so that's everything that I want to cover for single cell analysis. So the next lecture, we're going to move to proteomics and mass spectrometry.